In this video, we will discuss about the different kind of genetic engineering that can be done in the NK cells so that this NK CAR based treatment can be improved or its efficiencies can be improved. So we will start with this panel A that is on the top left side. So in this case, it has shown that in the tumor microenvironment, there is a continuous increase in the immunosuppressive metabolites, and one of these metabolites could be uh, adenosine, and other things is that it can make the tumor microenvironment acidic. Tumors makes the tumor microenvironment acidic by releasing this lactic acid continuously, and this will reduce its pH. And because of this uh, immunosuppressive environment in the TME, the immune cells are not able to reach to the tumor cells. And in order to improve this NK cells so that it can able to reach the tumor cells and generate immune response, some of the approaches that has been shown here is first to inhibit this A2A receptors, which is the adenosine receptors. It will also act as a suppressive receptors. At the same time, high adenosine will upregulate this T regulatory cells, whose function is also to suppress the immune response. So in order to take care of this higher adenosine, these A2 CAR or A2A receptors can be disrupted or inhibited in the NK cells. And other things is that the NK cells should be make immunosuppressive towards the other cytokines that are being released, such as interleukin 10, TGF beta. So the TGF beta receptors can also be inhibited. So this kind of inhibitions can allow the NK cells to reach to the tumors in a more efficient manners. The other approach is this genetic disruptions of the immune checkpoint. So far, people who are familiar with this uh, channel understand that there is a checkpoint uh, that are been available in the immune cells, so that once those checkpoint receptors are being interacted by the tumor cells, it will deactivate or suppress the immune response. And some of the common checkpoint inhibitors are these PD-1 receptors that are usually expressed on these um, NK cells and also on the other immune cells. So here the example shown here the example shown is of this TIGIT and NKG2A. So when the tumor cells interact with these receptors, it will suppress the immune response. So one of the ways that can be done to improve its efficiencies and prevent these suppressions is by inhibiting these receptors. So by inhibiting these receptors will prevent the suppressions of these NK cells through these receptors. And the one of the ways, this is the genetic disruptions of this immune checkpoint. The other things is that intercellular signalings can also can be disrupted. Instead of disrupting the receptors, the intercellular signalings can be disrupted, and these things has been done in the laboratory-based experiment by inhibiting the CIS and thereby prevent the intercellular signaling activations to suppress the immune response. And in terms of the D, here what can be done is the overexpressions or ectopic expressions of these chemokine receptors, such as CXCR2 or CXCR4, and when these are overexpressed, then the cytokines that are released by the TME or the cell nearby these tumors will become sensitive. Let me re-explain this. So by ectopic expressions or overexpressions of some of the receptors such as CXCR2 and CXCR4, they are becoming more sensitive to the cytokines that are being released in the tumor microenvironment or nearby tumor cells. And once these NK cells become more sensitive towards those cytokines, it can easily be activated and can generate the immune response against that tumor microenvironment. And similar to this way, one things can be done is by this disrupting these glucocorticoid receptors. And these glucocorticoid receptors, by disrupting these receptors, it will prevent the NK cells suppressions. And this is kind of similar to what we have discussed in this metabolic reprogramming. So here the metabolic reprogramming is being disrupted, and here the receptors are being disrupted. Although genetic engineering by is been done in a way that receptors are not being expressed on the cell surface. So this is the difference, or we can say that a cellular part is being disrupted. And the final approach that is shown here is by preventing these CD8 activations, or one of the ways to do that is knock out the CD38 expressions on the NK cells. And by doing that, the immune suppressions will be prevented. Immune suppressions of these NK cells will be prevented. And this, so one of the ways to do that is by knocking down CD38, or by using this anti-CD38 antibodies with NK cells, so that this anti-CD38 will be covered by the anti-CD38 antibodies, and thereby preventing this preventing the suppressions of these NK cells. And all these approach mainly prevent the suppression of the NK cells, and at the same time increase the trafficking of the NK cells to the tumor sites. So this, by preventing this, inhibiting this CD38, it will prevent the suppressions of the NK cells in the TME, and allow the trafficking of these NK cells to the tumor sites, and generate immune response against the tumors. So these are some of the genetic engineering approaches that can be used, or in some cases these are even being used and are at clinical trial stage for this NK CAR based treatment against the tumors. So I hope you understood to some extent how these genetic engineering ideas are being used, or what are the genetic engineering based ideas that can be used to improve this NK CAR based treatment efficiencies. That's all for this video. If you need to understand more about their approaches in detail, you can go through the reference that has been provided in the description. Thank you for your time.